few months later, once you get all of these results, you compile them all into a beautiful report, and then you go into non-human primates. You go into macaques and chimps, and they're much more like humans. So that's where most potential drugs kind of are made or broken. And finally, you go into clinical trials and clinical studies. So this is really, you asked me what a day-to-day is, but this is like the journey of how a drug is developed. For me, it's basically depends on which part of the drug development journey we're in. If I'm in the design phase, I'll be designing a lot of molecules on the computer. If I'm in the testing phase, then I would have got these in-house and be testing them in a test tube or with cells. And then there are teams in the lab that will work on animals and so on. So different projects are always at different stages of completion, and we'll be working on those as priority comes up.
Okay. So all of these are kind of, there's a foundational level of where you're beginning this off, because I'm trying to understand, okay, you have to design experiments and then do it. But before that, what is the point at which this starts, in the sense that
So
how do
that is
you
hypothesis generation based on the disease, right? So then you have these particular sets of diseases. It's hard to explain it without breaching confidentiality of what diseases we're working on or
Fair
what
enough.
targets.
Fair enough.
But basically, you would have a few RNA targets, protein targets, some macromolecule that's in your body that is dysregulated in disease, and then you want to find a way to make it more like normal. So you have a disease protein, you want to make it a normal protein, or you have a aberrant production of that protein, and you want to fix the machinery that actually enables production of that protein, and so on. So that's where it starts.